All right, let's talk about something we probably all learned back in biology class. Genotype and phenotype. It seems so basic, right? So black and white. But what if I told you that the simple definition we all memorized isn't really the truth? That the real story is way more surprising.
You know the drill. Genotype is the genetic blueprint, the DNA you get from your parents. And the phenotype, that's what you can actually see and measure. Your eye color, your height, all that stuff. The genes for blue eyes are the genotype. Your actual blue eyes are the phenotype. Easy, right?
But come on, is it ever really that simple? Is that clean line between gene and trait a fundamental law of the universe, or is it, well, something we kind of made up?
To figure this out, we actually have to go way back. We have to go back to the guy who invented these words in the first place.
Meet Wilhelm Johansen. It's 1911, and he's a botanist working with beans. Now, Johansen was on a mission. He was tired of all the vague, fuzzy ideas about heredity and ancestral influences. He wanted to make genetics an exact science, something you could get cold, hard, repeatable results from.
And this, right here, is where things get really interesting, because his original idea is totally different from what we think of today. For Johansen, a genotype wasn't a piece of DNA. Not at all. It was a whole class of organisms, like an entire line of inbred beings that were all genetically the same. See, his goal wasn't to pinpoint a specific gene for a specific trait. His goal was to get repeatable results. He needed a stable, predictable group to experiment on.
And you can see it right here in his own words. He says, phenotypes are real things. You can see them, you can measure them. But the genotype? That was a concept. It was his intellectual tool for grouping his beings together to make the science work. It was an abstraction. So his whole project, his whole reason for inventing these terms was to get rid of the messiness. He wanted to escape the unpredictable chaos of nature and build a system where he could get predictable results every single time. And to do that, he needed one thing above all else. He needed the power of control. And how he achieved that was, frankly, revolutionary.
He and Gregor Mendel before him, they created this incredibly powerful system. First, you create pure lines of plants by inbreeding them until they are genetically identical. Then, you grow all of them in perfectly uniform conditions. Same soil, same water, same light. It's only after you've eliminated all that other variation that you make your controlled crosses. And voila, that's when you see those simple, beautiful, predictable ratios, like the famous 3:1.
So just think about what's really happening here. The magic of this method is that you are systematically stripping away all the messy complexity of the real world. You're getting rid of genetic diversity. You're getting rid of environmental changes. You're focusing only on simple either-or traits. The whole thing only works because everything else is held perfectly still. The secret ingredient, the key to the whole thing, was control. Absolute, rigorous, experimental control.
And here it is, the main point of this whole explainer. That clean, simple, one-to-one link between a gene and a trait, it isn't some fundamental truth we discovered out in nature. It's an outcome of this process. It's a relationship that only appears, that only becomes visible, inside the highly artificial world of the lab. We didn't so much find it as we created the conditions for it to exist.
So, okay, if this idea was born in a sterile lab, what in the world happens when we try to take it back out into messy reality?
This is the million-dollar question, the massive challenge that biology has been wrestling with for over a century. How do you take all that complexity you so carefully removed and put it all back in?
Because the real world is nothing like that lab. Out here, we don't have inbred lines. We have genetically diverse, mixed-up populations. We don't have uniform conditions. We have constantly changing, chaotic environments. And those simple, predictable traits, in the real world, they become messy, continuous spectrums of variation. It's a whole different ballgame.
So you see the paradox, right? The very method that let us see heredity with such amazing clarity. Well, it did that by leaving behind all the complexity it was supposed to be explaining in the first place. It gave us a clear answer by ignoring most of the question. Now, this isn't just some quirky fact of science history. This way of thinking, born in a lab, it has had a huge, profound impact on how we see the world.
That distinction we started with, it isn't just a definition. It's not neutral. It's an idea that's loaded with meaning. It signifies a whole bundle of assumptions about how life actually works. And look at the powerful ideas that just tumble out from this simple distinction. It implies this hard wall between what you experience in life and what you pass on. It helps cement the idea that evolution is just a numbers game of gene frequencies.
It makes it seem like development is this simple one-way street from DNA to you. And it even suggests that the real you is the gene, hidden away, and your body is just its temporary expression.
It even shapes how we talk about the famous nature-nurture debate. We talk about it as if it's one single question. But in reality, it's a bunch of different sciences asking different questions in different ways. And sometimes that simple lab-born idea of genotype versus phenotype can actually get in the way of understanding the incredible complexity they're all trying to figure out.
This quote from the source material just hits the nail on the head. Experimental control doesn't just let you see things clearly, it also excludes many factors from being seen to make a difference. Our tools don't just reveal the world, they actively shape what parts of the world we're even able to see.
And that leaves us with one last big provocative thought. We started with what we thought was a simple biological fact, but we ended up questioning how we even produce scientific knowledge itself. If one of the most fundamental ideas in biology was actually shaped by the artificial limits of a lab experiment, it makes you wonder, doesn't it? What other truths out there are really just reflections of the tools we use to find them?